Boy, a big difference at 600 meters and 3,000, how fast Atalanta moves. Yeah.
We are at waypoint 2, and towards the, I don't know, 1/4 the way up the seamount, so we'll be heading uphill to waypoint 3.
Okay.
And yeah, so if we see anything interesting, we'll just stop on the way. But our heading to waypoint 3 currently is 005.
Yeah, I think, so I expect the sponges and a few corals to be the most common animals we see here, but it would be good to keep an eye on things that look different. And also, I would like to sample a few of the sponges. Obviously, some of them are quite large, so we'll have to pick things that are not too large. But we've done a reasonable job at sampling some of the corals, but the sponges are something we haven't been able to sample so much. So I want to make sure that we make good effort in capturing the diversity.
Just going through my ballast check here.
Yep.
So what should we be looking out for in terms of diversity of sponges?
I think mainly different shapes, different morphologies, right? So keep an eye on the different kinds that we see.
I see. So we have these like puffy cloud shapes.
Puffy clouds. And there'll be some like more like bases or more like some won't have stalks.
Cool.
Color wouldn't be that good of an indicator. I mean, that'll be more an indicator of whether they're alive or dead.
I see.
It's more the shapes.
Mm-hmm. So shapes are speaking to different
species.
Species, yeah. Okay. So
That's
they're
right.
not different morphologies of the same species.
Well,
then.
some could be. I think most of the sponges haven't been studied enough to,
okay.
to tell. Yeah.
Okay, gotcha.
Are most sponges like fiberglassy?
Yeah, so these are all glass sponges. So they have spicules made of silica. And the stalks very much look like fiberglass fibers. So
that's interesting because, like, you know, in sci-fi they always talk about like alternate forms of life in other planets, and they might talk about being silica-based. Even though I know sponges aren't necessarily silica-based, they just have produced silica structures. Right?
that's right. Yeah, so glass sponges are the most common kind of sponge in the deep sea, or that class of sponges.
All right, I'm going to start heading over towards the waypoint.
Copy.
Would
Be
you,
heading 005.
I was going to say, would you give the ship another 15 meters back in that direction?
Towards waypoint 2?
Are we going?
No, we don't. Santiago, did you need to go directly over waypoint 2? Or are we good from here to go towards 3?
Yeah, those are just indicators. There's nothing special about waypoint 2.
Yeah.
Right.
Okay.
Range
So
and
we can
bearing to wherever we're going?
005
005.
is the heading.
I'll follow you over.
And it's 430
please.
meters.
So as you go over, I'll start change my heading.
I'm with you.
So at what point are we going to leave where you've seen before? Like how long until we're in totally new?
Oh, we started on a different side of the
Oh, we're on a
seamount.
different side of the seamount
Yeah.
entirely. Oh, okay.
So this is all new.
Cool.
Yeah, so we're starting from this is a southern facing ridge, but we're in the southeastern part of the seamount. The previous dive was on the northeastern side of the seamount.
Correct.
Oh, that's interesting. You see that
Love the color of the
line, like
stem.
distinct line from one section of rock to other.
Yeah, so
Mm.
the fault there.
Cool.
So this is pretty typical, this kind of seamounts. You don't see too many loose rocks. You see that
I really like how clear it
everything's cemented
Yeah.
Mhm.
in place by
That's why
iron
we
manganese.
stopping to pick up rocks. No picking up rocks.
Well, I think we want to, but it's challenging. As you can see, everything's kind of glued together by the crust.
Oh, there's another of those fault lines or whatever.
Yeah.
I really like this configuration.
Cool.
That way the tether's out of the way.
Just letting Herc get ahead a little, then I'll start moving the ship.
What's that?
We can start
Probably
with
start moving the ship soon.
Sure.
No.
And once again, we're going 005.
005.
I think we can start moving now because it's going to take a while
Bridge nav.
for the swing to happen.
These are definitely giving
Step
Dr. Seuss
30
vibes.
meters,
These,
heading
these
005.
sponges.
Yeah.
They look like truffalo trees or...
So what's really cool, these faults look pretty old, so I don't think this would cause them, but along the Tonga Trench, kind of where American Samoa is, there's this thing called slab rollback that occurs kind of in the subduction zone where you get some plate flexure away from the trench, further in on the plate, and it can cause rifts and fractures that enhance volcanism. So again, I'm not sure where these are, the origin of these faults, but just kind of made me think of that.
Cool. Yeah, they're definitely really
Shana,
neat
would
to
you
see.
go ahead and push?
Well, yeah, what's the wrinkles? Is it a dead sponge or some wrinkle lava?
Ship's on the move.
Ooh.
That's very interesting.
What do you think that is? Dirty towel.
That's what I
It's-
thought too, Gabby.
I think it's a dead sponge.
So it looks like it's got some corals in it too, the little sticks.
Man, I want to poke it.
Yeah, it looks like it was one big sponge at one point. Or
That'll
maybe
be my
a couple
guess,
slushes.
yeah.
But because there's that stock right there, that looks like a stock.
Right,
And then
right.
I think there was one on the other side as well. So
All right, let's go ahead
Yeah.
and pull.
I think there's a lot of dead sponges
Oh.
around. You see the stalks on
Yep.
the left.
I'm a marine biologist now.
Yeah.
Yeah. Cool.
You see that.
And those are large sponges too. I'm just looking at the two dots.
Yeah, they're very large.
So that's 2 dots are 10 centimeters, and so
You can see.
That looks like a different kind of sponge back
Yeah,
there.
this one looks a bit different. Maybe we can take a look at that, please.
That one looks like a wobbuffet.
Yes, it does.
And how fast do these sponges grow? So how old would they be?
I'm not sure we know about the
Yeah.
specific sponges, but glass sponges some glass sponges have been aged to be the oldest animals alive, so up to 10,000 years, a single sponge. So they can be quite old. Oh, there's a bamboo whip.
It's a bamboo.
oh
Okay.
wow. Yeah, I think it's amazing when you realize the scale of these from those 2 dots, how big these are, how tall, and that sponge, it's big.
Yeah.
That's right. Yeah, that sponge is like 50 centimeters across and like
Yeah.
easy a meter tall.
It's interesting with the color of their stem.
Yeah.
Yeah.
I typically, think of green as like, photosynthesis. And
Right.
it's clearly not getting that down here, but
No, it must be some sort of pigment.
yeah,
That's
pigmentation,
Yeah,
turning that green.
but it helps, you can see the stalks of dead sponges there. Obviously, they don't have that color, so it's an indicator of
Yeah.
living tissue too. You see there's a much old, you guys are like more recent debt, and this one is a much older debt. And then we see some others that are, we'll probably see some that are fossilized in place. So this looks like a different, like the other one we saw. Tito, can we get a closer look at this one?
Sure.
Please?
Would you come down just a few meters?
Copy.
And Jenny, you can go ahead and push. So might this be a wetting sponge?
Wetting sponge?
We're the one kind that 2 shrimp get stuck inside?
Oh, I don't think so. There are also glass sponges, but those have a different morphology. More like they have a lot of open spaces inside. This one looks like a much more densely packed tissue, so I wouldn't rule out that there are little animals in there, but they'll be very small.
Ships in hold.
Have any of these kinds of sponges been successfully grown in a lab? Like,
I don't think so.
Yeah, they
No.
can only be in the environment.
Yeah, really things that are animals that live in the deep sea are very, very hard to keep alive, even just to keep alive.
What about like deep sea corals? Have there been any that have been successfully kept in a lab environment?
Yes, but not from very deep.
Oh.
So less than 1,000 meters,
Now,
usually
would you pull out
less
just a
than
bit?
600 meters is,
Gonna
you have better
go
success.
to the right and up.
Oh, there's some fossilized sponges there.
There was some other stuff right up
that's
Oh,
here.
brittle stars. They're the greatest.
Right.
Yeah, there's brittle stars that
Go ahead
colonize
and
the
push.
bed stalks. That's a different kind of sponge.
Does that look like it's decaying or?
Yeah, I was
Okay.
wondering the same. It has a brownish coloration, and you can see some of the skeleton, the silica skeleton, kind of poking out on the edges. So I'm not sure it's doing great.
And let's go ahead and pull, please.
We'll see. We see some of similar morphology that are maybe more white.
Oh, do you want to do any sampling here?
Yeah, I was thinking of trying to sample this one.
How much
Sure. Will that be challenging to get it to sink into a box?
Probably.
So where would you suggest we put that so that we don't have to, well, what would be the least impact on the rest of the dive?
What are we trying to sample?
That sponge.
Do you want the whole thing or just a piece?
Is that something we can suck up?
If we could suck up parts of it, maybe, like kind of cut it off.
I'm going to guess they're looking for the whole thing. Is that correct, Santiago?
I think we could do with a piece, but I think we will squeeze it. You know, when you manipulate it, it'll be. You could probably break apart. So maybe if you can get like the top half.
Sure.
How big? Can you turn on the lasers again, please?
Lasers.
Oh, they're on. So that's like what, 30 centimeters tall? Yeah, I would say if you can grab a piece from the top, maybe like a aim for like.
Would it be possible to get the? Do you think we should get the?
Yeah, that
So
top
I'm going
half.
to go with 35 high and 15 wide.
Yeah. Let's go with like 15 by 15.
What's the nicest thing to do to the sponge? Take the whole thing or just half?
I think half. It'll probably
Okay.
regenerate.
Do you,
All right,
do you
I'm
want to
gonna-
bring the ship back? Like
There's some
5,
classic
maybe?
experiments with people
You know, I'm thinking you
just
just might have
associate
to turn
all
a
the
little
cells
bit.
of the sponge and they'll like
Atalanta's
come
going to end
together
up over
again.
here,
Cool.
and I don't think the
I don't know if
distance
these
will be
ones
too
do that,
much.
but
Okay.
We don't
could
know
bring
much
the
about
ship,
these sponges. So
10 meters ahead and 10 meters south. That wouldn't hurt anything.
You want the ship 10 meters south.
10 meters east and 10 meters south. So how's about 15 meters at 135?
Copy.
And tilting down.
Bridge nav. Can we step 15 meters, heading 135?
Oh, once again, Santiago, where would you like to put this sample?
Oh my gosh, man. Let's try a starboard bio box. I'm just thinking, looking at all the other sponge remains, it seems like they stay in place easy, so...
Oh, yeah. Yeah, they seem to, even the ones that fell over don't seem to be bobbing around.
Right, so I'm gonna go with that as possible evidence, that they are negative buoyant, and then won't try to
If you want in
float.
the starboard, when you land,
Oh, that
all secure.
sponge in the back, we see a different perspective.
Oh yeah.
I'll use a little extra thrust.
Oh yeah, you can see these two may
Oh,
have.
both of them. They're,
Yeah,
it's
have
like
similar
cup-shaped.
morphology. So I wonder if the larger one is just a larger individual that is not alive or not doing well.
Yeah.
When you're situated.
Cool.
There you go, 100% joy gain, 100% down. You can secure
Oh,
the
that's actually a
starboard
neat
thruster.
view.
And there's another kind of sponge here. You see it?
Starboard
Securing-
vertical.
Uh-huh.
That might also be a good
Securing
sample because
starboard
it's not
bridge.
too large.
Looks like a pig snout.
That looks good.
Jacob, if you see along the way
let's
a
bring
rock
the
that
box
looks
all
good
the way
to you,
open.
feel free to say so.
Okay.
Yeah.
Haven't seen any.
I'll go ahead
You're
and
on
open the
rock
box
watch.
already.
Rock watch, yeah.
Let me go ahead and go into sample mode. Actually, I take that back. I'm gonna go stay here until you get your position.
And I believe Atalanta's going to go kind of,
So let's say we sampled this guy.
should settle
and
almost on top.
Find out,
yeah.
oops, it wasn't alive. Well, would it be, is it still a good sample? Like,
Yeah, I mean, this looks very much alive.
Okay.
Yeah. Can we get a close-up before sampling? I just want to see what...
Yeah,
No.
let me just get a couple things powered up.
No
Yeah.
close-ups.
There's a few
And
critters on this sponge.
panning right. And tilting up. Go ahead, push.
cool. Itty bitty brittle stars on it too.
Yeah, there's some brittle stars. There's something on the right. It could be like a...
Oh yeah.
it's like a shrimp or
Is
a
that some
swallow.
coral in there, or
Are
those
those
the little
hydroids
star?
on the right?
On Atalanta.
This is a crinoid.
Hydroids on the right?
Try to get her.
I don't see what you mean. This is some... might be like a shrimp. Looks like a shrimp, yeah. Offroids, where do you see hydroids in it?
I don't know if that's what it is.
Oh wait, I got
Like-
it.
Can you...
On the
Perfect.
Describe.
Look at that.
on the right, yeah, sort of moved towards
No.
the center now.
to the right of the sponge or on the sponge?
On the sponge, yeah.
That's a crinoid.
Oh, that's a crinoid. Okay,
Yeah.
I just saw sort of the feathery.
Yeah.
Right in the center of the
And that
screen
was-
there, that's
yeah.
a crinoid.
I think so,
Okay.
yeah. You can see the appendages that it's using to like
Okay.
hold on to the sponge.
And that's a shrimp,
Yeah.
I assume. Yeah.
Yeah, it is a shrimp. Cool, thank you.
So from this vantage point, I also am looking at, over on this screen, that little sponge that you were looking at.
Yeah,
On here, it kind of looks like it's a miniature version of what's on the stalk up back behind it on the bigger side.
that's right, yeah. That's what I was thinking of, would be a good
Cool.
target.
Here we go.
No, we didn't put the coral cutters. Oh well. Oh yeah, we did.
Yeah, I'm going to try and cut it right in the middle.
You can zoom in if you want, if it's doable. But
Santiago?
Yeah, maybe a little bit higher.
Okay, I'm not sure the blades are wide enough, and that'll end up being tearing. I was kind of going towards the taper, where it tapers down a little.
Yeah, you probably break off easy, that's my guess.
Sure.
Like
okay.
once it's
Or tear.
tear or
So
something.
is that high enough right there?
Yeah, that's good.
Got a really cool shot in Atalanta.
Yeah.
Ooh, yeah. With the firebox open, that's really cool.
Oops, I'm pushing on a rock, so that's... a second.
Copy. I'm gonna see if I can get the.
I'm just pushing on a rock. I can't get the blades. So
I got bubble on you over here as well, so you can kind of see behind the rock.
yeah, and see how it's right up against the rock?
Yeah.
So the blades are inches up. So I'm just going to go in right there.
Can you Oh. Looks like you got it.
Oh yeah.
Sorry, a little more than we expected, but
You want to zoom in before we put it in the box, or?
yeah. Do you want some pictures before the box, or?
Yes, that'll be
I'll
great.
go ahead. Do you want them with or without the lights? The lasers?
With the lasers.
Yeah, you can push.
Okay. Oh, I guess you can't really see the lasers on right now. Oh, and you got a little brittle.
Yeah, you got
All
it.
the
Bruce,
things.
All right, I'm going to try to put that in one of the small compartments.
Oh, this is my favorite part. Seeing it on Atalanta. All right, go ahead and sample them.
Yeah. That is some really good Atalanta view. Water's nice and clear.
Mm-hmm.
Oh no.
Oh, floating away.
Don't
Yeah,
float.
just taking right off.
Floats.
Yeah.
Sink or float.
It was floating. I'm
Through the-
sorry. I didn't think it was floating.
Usually, I usually don't see them float too much.
It definitely took off.
We had already secured the vertical thruster to kind of reduce the influence and all that, but
It was
yeah.
too floaty. Sorry about that.
Maybe we can slurp the
Got you
red,
right
what's
here.
left there?
You want to try slurping?
Yeah. Seems that otherwise it would be really hard to get in a bio box. And even if we do in the front one, it might float away when we open it to get something else in there.
All right. If you want to try slurping, let me switch cameras real quick.
That would be a good, you know, we've had all these Lego robotics teams asking, like, what's a challenge? What's an obstacle? Something to weigh down samples, keep the bio box
Yeah,
from.
I know.
There you go.
We
There's
have
one.
some-
So I think what I'd like to do is just grab the rest of it and put it in the slurp.
That sounds good.
All right, I'm flushing. And since we haven't done anything, I'll go ahead and go to... What number is that? Number 1?
One.
Number 1.
Let me know if you need any camera changes. There you go, I got you on bubble as well. Got it, all right.
I'm pausing the sampler so we don't get the sediments.
Okay.
Pan down a bit. Are you ready?
Yep.
then I think on bubble you'll have a good view on.
Yeah, so this will still
where the
be a good
suction
sample
is.
because one of the main ways that
I can't see much.
taxonomists
I'll be paying that
identify the sponges is by looking in the microscope at the spicules. They have the very unique morphologies that they
You
use
see
to
it better or
distinguish
no?
species.
Do we have a microscope in the wet lab?
I don't think so.
There it is.
I haven't seen one. I asked that early on the cruise and
And
can't
I'm
remember.
gonna go ahead and
It
start.
seemed like people
Pumping.
weren't sure, but most likely not. But this is the kind of thing that you want to use an electron microscope
Got it.
to get a good image with to appreciate the three-dimensional
I see.
complexity of the structures.
That seems really there, right? Would you agree that seems like the spot?
That seems pretty close, yeah.
All right, 100% slurp.
I am at a hundo.
I'm trying to,
Feather it out.
oopsie. Slurping 100%.
Yes.
Doesn't seem to be pulling it.
I think it's angled down. Angled down towards the arm. Like you're right on top of it.
Okay, you're slurping.
Yeah, it should be. No. Well, it's there. Maybe it needs a couple jiggles on the. What's in the thing?
My money don't jiggle jiggle.
Oh, looks like it's like just too big. There it goes. You got it.
Good job.
Thank you.
Boom. There it
Yep.
is.
Nicely done.
Nice.
Slurp.
Excellent. Well, now we have a strategy for the sponges.
There it is.
Yeah, on the old one we used to just scrape and slurp. It was made out of metal and it was really sharp, so it was really nice. This one's a little bit harder because it's a plastic, but it should still do the job, I think. If we in a pinch. All right, so that's in number 1.
Yeah, and then if you pan up before moving, that's another target for collection. We might have to reposition, but I just wanted to
Jacob, what number?
point it up.
212. I forgot to tell you about 211, which is the eDNA pump.
Okay, thank you.
Are we moving, or do you want to get something
No, I
else?
want to, if you can pan up, I wanted to see another sponge that I think will be good for sampling. You see this small one here?
That little guy.
So it looks like it
Could
could
you
be
do
one,
one more time? Circle that.
Yeah, this one.
And we're going to slurp that.
Yes.
All right. Gonna
That seems like a good size for a sample compared to the bigger ones in the back.
can I have the starboard vertical back, please?
You have it.
Some fossilized sponges down in there.
Mhm.
Do quote-unquote fossilized sponges tend to
Nope.
be just as sturdy, or are they brittle, or?
I don't know. I was just thinking
Oh.
that we should sample one. I don't think we've ever sampled one of those. We saw them in 2017. We've never sampled one. I don't. It would be really interesting to get a sample of those. Maybe we can get a couple pieces from this pile. Just curious about how thick that manganese crust is,
Mm-hmm.
and like.
Yeah. Oop.
Oh no.
Oh no, what?
The
Update.
computer restarted.
That's all right. Let's just watch our delta.
We will.
Graph valves. I'm sure it'll be updated by the end of the dive.
It's funny.
Thank you, Windows.
Oh, don't you love it when your computer just decides to?
Update on its own.
Yeah, it just doesn't
Hey,
matter what you're doing.
sometimes, hey, I need a break too. You know what I mean?
Yeah, if anybody has an important computer that they don't want to restart, I would pause your updates because that's an automatic one that happened today. So
Oh,
I
okay.
paused mine for today. So check your automatic updates if you don't want it to do that today.
Mm.
I even told it not now. Do it on your own time.
Reminds me of an episode of, what's that, that Space Force show?
Space
Pretty
Force?
sure- yeah, they had the funny show where it's like Windows Update and they're doing a launch.
Space shuttle? Or-
Yeah, they're like, no.
All right, as soon as I
That would be our luck.
get this slurp set, I may have you basket back in.
Hold on. I need to get to a camera, and then I also need to flush it out and stuff. Okay.
Well, that AC's up and running again.
Yeah,
Yeah.
Winter has arrived.
it was nice while it lasted.
Okay, basket back in, please.
Okay. You want the porch back in?
Porch back in, please.
Where'd it go?
Just
Porch in. There you go.
so I could use a view of that a little better, but let's go ahead and start slurping.
Let me get to the box.
We don't want it in the, yeah, there you go, number 2.
What's that?
I need to change buckets,
He's got it.
but I have it now. So, all right, starting the suction slurper. It is slurping. We got it.
There it goes. Very nice.
Like a little piece of cauliflower.
Did you say you wanted some of the fossilized too?
Yeah.
And did you want it in the same bucket or no?
Maybe
Might.
that one we just grab and put in the bio box or in the...
That's
I'm looking at what's under the porch now.
Would you like to circle what you'd like for a sample?
Yeah, I don't see it right now. It's like under the porch. Is there any chance you can...
All right, I had set up for slurping. I can, with the manipulator, do a bit more. So let me go ahead and freeze the manip and
Thanks.
we'll back out a little bit. And I have all my thrusters.
yes.
I do, yeah.
Oh my god, do you know the sonar? Is there a password? Oh, never mind, I know what it is.
Jacob, is that a new number?
Yep, that is 213.
213, thank you.
Santiago, are you seeing what you're looking for now?
Yes, this...
Oops.
See that? It looks like a
Looks like a vertebrae.
vertebrate, yeah, like a backbone.
It's CNET, right?
Oh, it is pretty though.
Crumbly.
That doesn't seem like it's going to hold together for
I think it's the Kongsberg one. The red box.
This one?
I think so.
Aha! What is this? This is 165? I don't know what any of this stuff is.
You got it.
Nice.
I'm gonna go with default.
Very gentle.
where are we going?
Okay.
Well, I was thinking to
Um,
start with bio box, but I'm
try
afraid it
a
will
heading run.
float away.
Oh, I guess I could do it here.
But it has the cross that are... I don't know.
Starboard bio box out, please.
Yeah, let's
Copy.
try it. Another experiment.
We'll see.
And I'm gonna go, as you come about, I will secure the vert.
Roger that. Go ahead and secure the vert.
Securing vert.
Might see a little wiggle, but it shouldn't take off.
Oh, cool, we got it? We got
Thanks, Michael. That was easier than expected.
it. Yeah.
Okay, would you give me the starboard bio box sampling salvo?
Awesome. Thank you. I'm very curious about that.
Yeah.
We'd be interested in see if sponge
I'm
taxonomist
sorry.
can
Used to
identify the
have
sponge
access
from
to
the
there.
Yeah.
I had a viewer a little bit ago, and I say that, so when they look at the spicules, they dissolve away the organic part of the tissue.
That's right.
So is that possible still with these kind of fossilized sponge parts?
That's what I think.
Cool.
Yeah, that's what I was curious too. Maybe able to identify.
The jaws are thin enough so they can
What was
fit
it when
in
it was
the
living,
boxes
right?
that we need to get in there.
Or
What's that?
like if
The
it's been dead
jaws
for a long time.
are slim enough,
Right.
And it's-
so if you
that'd be
needed
cool.
to get in the
Yeah.
box, you can.
Yeah, so when they open, they're much wider than the box. Right inside.
Closing box.
And that went into the second one from the aft portion.
Dang it, I keep forgetting to relabel those boxes. All right.
All right.
And I'm re-engaging starboard vert, so you'll get a bump.
Thank you.
And that is sample 214.
214.
Anything else while we're here?
Did he leave?
Yeah, he's oh, there it is.
Well, waypoint 3 is directly north, 000.
I'm resuming the pump.
Great, thank you so much for the samples. Very good.
So getting a little late in the cruise. I'm wondering, do you want to try flying for 10 minutes?
What's that?
Would you like to try flying for 10 minutes?
I don't know.
Say yes.
Yes, I suppose so.
Go for it.
Super. All right.
Do some donuts.
Okay. Just all right, so there's, can you hear me?
Yes.
Yeah. So right now I've got it in auto XY
Yep.
and auto depth, which means that it's going to maintain depending on how we have it set up here. So right now we have it in auto depth, which means it's using the Doppler or the altimeter. I couldn't tell you which. If we went to auto altitude, it would maintain its depth. Excuse me, it would maintain its height. So I think we're going to switch over to auto altitude and see if anything happens. That's pretty good. We're about 3 meters up right now. So the stick, forward, backwards, sideways, sideways, turn the heading,
Oh, cool.
right? This is pull up, push down, which is just like an airplane, right?
Yep.
Okay, so a lot of people are unintuitively trying to go up, trying to go down, which is the opposite of how the stick works.
Gotcha.
So all we're going to do is shut off auto XY, and then we're going to start flying the camera. I've left a little bit of the basket in view to try and give you a bit of a reference.
Copy.
You don't have anything to give you 3D, so it's
Okay.
you kind of use your intuition. Try not to use try to use that camera up there a little bit to kind of see when you might hit the bottom,
Okay.
but there's not much of a shadow and all that. But I would say press the auto XY button, leave the stick in your left hand alone, and just start driving around with that one. The first thing you're going to do is kind of shift off to the right before you even start moving. No,
And
I'll
bump
stop.
the rock a little bit.
Yeah, no worries. But I'll stop. Rotate your heading to north, because that's the direction we're going to be going.
Okay.
And you're in a good position in Atalanta, so that's a good thing. But we're, yep, keep going. So you're going to rotate to the north. So you're going to twist the top of the stick. Let go of the stick. Rotate the top and watch your heading change right here. So you're going to go to due north.
Copy that.
And without getting too far ahead, once you start driving ahead, but what I like to do to kind of keep as much of the bottom in sight as we can is lateral left and lateral right. And we're about 3.5 meters up, so you should be okay as far as hitting the bottom. If you feel like you're going to hit the bottom, grab the stick and pull. And you're not actually moving what you're doing is moving the set point because you're in auto depth. So as long as you pull on it, pull 100%, it's going to come up as much as the velocity allows it to, which our max V is 1 meter per second.
Ooh.
Yeah.
1 meter per
Don't
second.
fall out of your chair. Would you like some armrests? I find
sure.
it really nice
Thank
to
you.
rest your arms.
All right.
And then I guess you can just call in the move when we're ready to move.
Yeah, I'm going to call the
do-
bridge and ask them to go 30 meters north.
Yep, 30 meters north.
30 meters, 0. Can I just say north? No,
Just use the
okay.
heading, just in case.
And let's see, bridge. And I just click up or click down?
Click down when you see the white. That's when you're talking.
Bridge control van. Yes, would you please go 30 meters, bearing 000. 30 meters, 000. Roger that, thank you. So now it's getting a little steep.
Yep.
You can see that. So I might come out of auto depth, and that's that button, yeah, and just use a little bit more of exactly.
You see that starfish.
Oh, yeah.
Right. Ooh, cool sea star there.
That's a cool starfish, sea star. Do you know what that is?
Sea star.
It's a sea star.
Oh, Michael, would you pull a little cable in?
Yep.
And focus on lateraling right, not driving ahead yet.
Copy.
Looks like we'll be going uphill in the next 20 to 30
Notice
meters.
that from your sonar.
I'm gonna jump in interaction for a couple of minutes, but I'll be back. You guys can keep heading on that move and seeing
Drive
what
around
else
the
you
parking
see.
lot.
Yeah.
Copy.
We'll be gone by the time you get back. He's gone, go.
So even if I'm steering right, it's not really going right too much. Am I in a,
So lateral doesn't have a
or
lot
sorry,
of-
left?
It takes, you've got to hold it 100% for a while. They're the smallest thrusters on the vehicle, and it just takes a little
wow.
bit. And it's also a hydraulic system, so it's not instantaneous.
Gotcha.
But it will get going. You just got to give it 100%, and I've got your joy gain as strong as it goes. So
Copy.
only seem slow if you're used to flying.
Yeah. That's funny.
And one of the first good pieces of advice I got when I was on my first ROV cruise was, if things are happening fast, you're doing it wrong. And if you find yourself unable to make way in the direction you want to go, just rotate the heading and drive that way.
Gotcha. That makes sense.
That's
But
also
don't
good.
get too far out ahead.
Yep.
And like I said, maybe lateral right.
Slow is safe and safe is fast.
And to get a feeling for how Atalanta's moving, what I'll do is sometimes put the cursor or the mouse on it and then just see what kind of progress it makes. But it's such a lag at 3,000 meters or 2,700 here. So if you see something that you think science might be interested in, try and get it into the center of the screen by rotating and that, and then ask Jaina for a push.
Let's have another sponge.
And now you can push down and, you know, land the vehicle.
Push down and land.
Yep.
Okay.
And then we will tilt the camera up as soon as you're on the ground. So we'll go to,
We're going
oops,
up.
you've got to hold it down to,
Yep. Does it land hard?
it's getting a little too far away.
Okay.
So when you land, you might want to kind of go forward and down at the same time.
Okay. There's another one.
You're doing great, Sweaty.
Ooh, I'm going to say that would be a good spot. So back up a little bit, back up a little. You don't want to get too close.
Right there, perfect. And then just kind of push down and then...
And hold the stick like that. So a little bit forward and a little down, and then you press that button I'm always asking you to help me with.
Which button?
It's either that
Oh,
one or
that
that
one.
one. Yep, right there. So now we're going to tilt up a little bit, and we're going to ask Janer to push.
Okay, Jaina, can you push?
Yeah, because when we do that, we're driving into the thing.
Okay. Yay, sponge.
It looks like we may have Atlanta moving, and it's starting to feel
Just
the
getting going.
swing.
Yeah.
Okay.
So no rush. We're here for a bit. And don't use Atlanta as we're zoomed in on Atlanta, so you don't need to worry.
Okay.
So you're a little more familiar with this.
Yes.
So that's a good reference.
So let's go ahead and pull back out. And so now what I do is I square up the camera as best I can, and I use the basket edge. So I'm going to come down, and then I'm going to pan right, and then I'm just going to tilt up a little bit. And go ahead and straight up. So out of stick lock, straight up, and try to fly over the sponge.
I'm just trying to see if I've cleared it.
You cleared it.
I would
Okay.
drive ahead just a wee bit. There's some more vertebrae from old sponges. Next time the vehicle's on deck, take a look at the laterals versus the verticals
Okay.
and the horizontals. Their horizontals and verticals are identical, and the laterals are these little
They're
tiny things.
I think the power is exponential, so it's like 3/4 of the size of the thrusters equals to a little bit more than 1/3 of the power.
And so I'm going to guess there's a little current that seems to go that way a lot
Yeah, it
easier than it
feels
goes
it,
that way.
for
So
sure.
it's just going to be a little bit of current kind of causing that.
Okay.
Jaina, you want to pop in for 10 minutes?
Yeah, Jaina.
All right, so hit X, Y, and depth again if you don't mind. And I'm going silent.
Yeah, I can do it from here. Oh, there's a button. Okay.
All right, Jaina, can you hear me?
Yes, I can hear you. Can you hear me?
Alrighty, I can. So right in front of you, you've got, right now, auto XY and auto depth on. The stick in your right hand is going to, any direction you push it is going to drive the vehicle. But when you want to change the heading of the vehicle or rotate it, you twist the top of it. So I was going to say, go ahead and click auto XY and start driving ahead. And nice and slow. And try to just moderate the stick because I've got the power at 100%.
Yep.
And if you need to, come out of auto depth and start using the stick in your left hand, pull up and push down. But you want to maintain a nice distance so that we can see the bottom well.
You're coming up.
Hey, would you get her to Face ID her phone so I can take a picture or two? Needs Face ID.
I don't know how to do
My
this.
face didn't work.
My face isn't working either.
I don't know how to do iPhones.
DJ, did you get one of me?
I
I
think so
didn't,
many
but I
of you,
saw
Gabby.
Jaina
I
did
have a
some.
million.
Oh, cool. Thanks.
Absolutely.
Proof. I'll put it on my resume now.
Yeah.
Oh, the front angle pictures.
Hey, would you glance this way? And one more time. There you go. I couldn't
Yeah, I
find the
just
camera
love the
on your
bright
phone.
color of the
I
stalks.
know.
So keep an eye on nav screen and
you
make
always
sure you're
wonder if they're
maintaining
as
a good
bright as they
distance
look
ahead of
here.
Atalanta.
Yeah, and also, you know, again, it's dark down there. So, what is the pigment serving, right? It may not be the way we, for visual purposes, may have some other properties.
This is a hype bee sponge in that fluorescent yellow. It's like
Sort of looks like, what does it look like? Cauliflower?
Yeah.
And you can see in the top one
It's
that
like
you're about to hit
cloud
that
factories.
other sponge,
Like
but
maybe
you're
they
coming
get
around it pretty
unsnipped
nice.
and
Bridge
rise.
now.
Yeah.
Maybe this is
So
where
that's good.
clouds come.
Yeah, there definitely feels
Can we
like
step 10 meters,
time.
heading 000?
Should we put a sign up, student driver?
Yeah.
I think it's on the back of Herc. You can see it in Atlanta. Don't mind my driving. But yeah, it's very much like I don't know if you've ever flown a drone. Very much like that.
Yeah, I mean, our
The response time's a little
Zeus
bit
camera, or not our Zeus camera, but our Bolton controller for all the cameras on deck is basically the same
different.
controls. Which is interesting that like all that different technology uses.
So I
the
would
same
suggest kind of
controls.
lateraling back and forth because we're getting a bit ahead of Atalanta. It will catch up, but it takes a while.
Okay.
So it was lateraling to the right's a lot easier than going left right now.
Yeah.
And then it's pretty flat, but those are obviously your rear views right over there. Those two at the
Okay.
top.
Yeah.
Yeah, very good. When you leave, do say it is the hardest thing ever, because
I'm back.
for our employment status.
Yeah.
Who were you guys interacting with?
Deep sea biology class at the University of Rhode Island. They were just learning about Lulu.
Yeah,
Oh.
so I'm
Okay.
trying to lateral
See
all the way
now,
left.
They were at the
Just
intersection.
give it 100% and leave it there.
Yeah.
Yeah.
Leave it there.
The current is pushing you.
Possibly.
Yeah, so
So
you're
it's
going
going to
against
take
the
10
current.
seconds and then it'll start moving.
Yeah.
So just watch an
Oh.
image in the camera, and that's the only way you're going to like
Jane
a
is fine
smaller
now.
one.
Very
But there
cool.
seems to be a little current pushing us to the east, so trying to go west is always going to be a little difficult.
Bridge nav.
And you can tell because of the tether, it's going
Can we
all
step
the way to the
10
east.
meters, heading 000?
So
Some really large sponges, but as you can see, it's mostly sponges. There's not too many other.
Been trying to look for some corals, but I haven't really seen many.
Yeah, we saw a couple of the just as we landed, but
how much do we have left on our move?
not too many. And this is, as I said, well, remember this is pretty typical of this depth, and on especially on the seamount.
And noticing, you know, we have little ones and big ones. We have, I guess they've had more time to grow and reproduce, and some get big, and as opposed to on the Viulu when they were all kind of the same size.
You guys look up more in this seat than I do. I feel like my neck is already sore.
Already?
Yeah, use this one. Don't use that big one.
Okay.
But you can also lean back. go into chill mode.
Go
Put
into-
put the armrests down.
I'm still
Slide.
in anxiety mode.
Yeah, you get some armrests. Use your armrests.
Ah.
And you get the other one on the
Oh,
other
that
side.
is nice. Okay.
Finally, we're getting some movement.
Yeah, watch the cup. But, yeah, hop right in. Hi, Jaina. I'm afraid our relief is here. Get up.
Okay, what buttons do I
So-
press?
Good job,
now?
Jaina.
Good job, Jaina. Good job, Gabby.
Gabby
Thank
is
you.
the, yeah,
It's like
nice
if I
job.
don't know.
Just leave
Yeah,
it
we can't have two Gabby ROVs though.
No, we can.
Is the
Thank
ship
you.
still moving, Gabby?
It's great. No, it's coming to a hold right now.
Okay,
This is perfect timing.
perfect timing, yeah.
Give us a minute to get through another shift change. And then the 4 to 8 watch will be signing off. Though you'll probably be sticking around, huh?
Maybe for a little while, then I might go to the lounge and watch from there. It's very cold now.
Oh. Oh, maybe the door. Yeah. How long is this dive set to be?
Um,
Oh, I'm saying 18 hours. So we've reached depth about 6 o'clock our time, 6 a.m. our time, and planning through at least a couple shift changes before
Yeah.
we come back up.
I think expected to be recovering around 9 p.m. our time.
Our time.
So that'll be about, so like a 16-hour dive.
Yeah.
yeah,
So if
probably.
you're just now tuning in, we are only at the beginning. Oh, yeah. If teachers, if you have kids that are going to recess and lunch, we will definitely still be in the water whenever you guys get back. So have a good lunch. We'll see you when you get back.
Yeah, we're just getting started, so this is going to be a long, fun dive.
I want to draw.
Yeah. But it does feel like warm air
So the-
again.
Hi, Miss Jones. We are so excited to have you coming out to sea with us very soon. We are right now in a dive of the Malulu Seamount here in the National Marine Sanctuary of American Samoa.
Yeah.
We are in a shift change, so we've got a little
Also,
bit
we
of,
have the,
you know, in and out and people kind of getting settled in their seats. But we've just started the dive a few hours ago, and we are excited to continue this for the next 14, 16 hours.
So we've already gotten two slurp samples and a fossilized sponge sample in the starboard vial.
Okay.
Good morning, Santiago.
Morning, Val.
What have you guys been seeing down here?
A lot of sponges.
What kinds?
Underwater
Glass sponges.
I saw Poliopogon before, and one of the stock sponges
Yeah.
before I went to
I think
bed
it's
last night.
Bolosoma or Califacus.
Okay. Yeah, I heard Megan say Califacus, but it was for the all-white one.
Yeah, the one that looks like.
Yeah.
Yeah, we
Okay.
sampled two.
Okay.
Two sponges and fossilized sponge skeleton,
Oh, cool.
and we're moving up slope to waypoint 2. So we started at 2750 meters. So we haven't moved too much. So we landed.
Pretty late.
There was a
Yeah.
delay in the recovery and redeployment. So we've been in the seafloor maybe for an hour and a half
Yeah.
or so.
Okay. And what kind of corals have we seen?
We've only really seen two corals, not in a while. We saw a bamboo whip, a long one, and then one of those ramuli gorges.
Oh.
The ones that are those are probably the most common corals, at least that.
What do those look like?
They look kind of like sea fans. They're white. But They have
Okay.
like bendy branches.
Okay.
They're completely white.
Okay.
Yeah, it is. And at the beginning of on the descent, it calms a couple times, but then that's about it. But I don't know why it's tricky. Yeah, I did once. I can do it again if you want. Yeah. You want the old, but then you don't get this one.
Can I give you a reset, Robert?
So I'm going to troubleshoot that CinemaCam jerky stuff.
And are you ready if I start the ship moving again? Keep us going? Okay.
Is the back of the van ready?
Yeah, we're ready to keep
To
going.
keep moving? Great.
Sorry, I thought you were talking to Robert.
Talking to everyone, but yeah.
We're ready.
Great. Bridge nav. Good morning. Can we have a ship move 30 meters due north, please? Thank you.
So you like the lowrider mode there,
Oh, that's a big dead sponge there.
huh? uh,
I think all this is a dead sponge.
Wow.
Oh. Is that
It's very large. You can see it on the overview camera,
yeah.
overhead camera.
And what type of sponge is that?
I don't know.
I thought you might have
Nothing like
an
we've
answer.
seen alive so far.
Oh,
Ooh, has tubes.
yeah, that is really interesting.
Yeah, this is what I remember from previous dives on this depth range. A lot of dead sponges and a lot of them that look nothing like the ones that are alive now.
that looks like it's been there a long time.
Yeah, it's getting that manganese coating already.
Would there be any actual tissue and DNA remaining, or is it too long dead for
I
that?
doubt it, yeah.
Looks like it's rock now.
Yeah.
Oh, there's a tiny sea star. One of those cookie sea stars.
Oh.
Can you, oh, shaky. Can you zoom out? Just curious to see how big this whole... Oh, you can see the base there.
Oh.
Whoa, that
Wow.
is really large. So that, if this is a whole, if it's one individual, it's several meters. Or it was several meters tall.
Almost
that
like a
was
small
huge.
tree.
But you can see the stalk right there where it was attached. It is huge. I mean, the base of that attachment is like 30 centimeters across.
Yeah.
Just I wonder if you can see where it was attached in the rock.
I wish we'd seen it alive. This is pretty impressive.
Yeah, well, maybe we will see something large like this. I mean, you see the live sponges right here,
Yeah,
really big.
quite large. Do you know what group those are, especially the big fluffy ones?
Big fluffy cows. Let me see. I think they're bolosoma, but that could be wrong. Let me check the guide.
You zoom in, Dave?
Look at
It's
that.
huge. It just looks like a big dead old tree.
It really does. Wow. Huh.
Hi.
zoom out.
Are you able to zoom on? There's a little coral behind the base
Yeah, I'm
of
on.
Okay.
the sponge we were looking
Very, very
at.
large sponge. We're just looking at this one that's dead,
It's falling
getting
over.
fossilized. It's like 2-3 meters tall. Well, it was.
Ferry-a-day sponge.
That's taller than
It's huge.
that. Like just
Yeah.
just the stump there is
Yeah, it's like
a
this.
meter.
Yeah, like
Yeah,
half
it's
a
like
meter.
the sequoias of sponges.
Yeah.
Here.
Yeah. So,
Do you think it's a fairy day?
it's a what?
I mean-
Ferry-a-day? I don't know.
That was the ones that we saw.
And look at how big that
Oh,
thing
you mean
is.
the family? Yeah, it could
Yeah.
be, yeah.
I think it was standing straight up like
We
that.
did sample 2 alive sponges already in this slurp, both of them.
Yep.
I think that's best way to sample them.
Yeah.
Here maybe see
what
a big one.
types were they?
There was one that looked like a concave shape cup, so not
on a stalk or
not
no?
on a stalk.
Okay.
And then we sampled a small one like those ones on a stalk, but it was like this big, the only
Okay.
small one we see, because all the other ones are massive. They're like 50,
Yeah, yeah.
60. I think there's some that are like a meter across here.
Holy cow.
Yeah, so I expect you to see a lot more sponges as you continue climbing uphill. We've only seen a couple corals, but mostly sponges.
What were the corals?
It was a bamboo whip.
Yep.
And one of those, well, like a fan, they're white. They call it a
Can you
muligorgia.
zoom in
Yeah.
again, Dave?
But they're very common. We've
Yeah.
sampled those before. So I was thinking that sampling a good diversity of the sponges here would be good.
Yep.
So we haven't really sampled too many sponges so far. And they're dominant here. What else? Something really interesting is that Mary and I noticed water column anomaly here around 560 meters. She saw it on the last dive when there was basically a cast.
Yep.
You see this blimp here?
This one?
Yeah, that
Yeah.
matches EH anomaly at the same depth.
Oh, interesting. It's not. Is it uncorrelated with oxygen?
Yeah, I was checking. The oxygen is a bit shallower. The oxygen minimum is around 400,
Okay.
450. So it's uncorrelated with that. But that's interesting.
Yeah.
I'll put the mapper to think of like getting some background data.
Where is this from? Mesobot or from
No, this is the previous dive,
the previous dive? Okay.
the bounce dive. We had to recover
Oh, interesting. Huh.
just now. This
Yeah,
is
yeah.
fresh data.
Oh, the freshest.
We're 30. The freshest was 30 miles away from Wailulu, but there's something going on in the water column.
Yeah, yeah. Huh.
Yeah.
Okay, what?
Oh,
There's a Remilagorgia. So we need a. Are you saying another couple weeks out here?
I think
We'll just
so.
stay.
Right.
Okay. Erica's like, no.
I'm kind of like, sign me up.
Well, at the very
That's
least, we want to
amazing.
reserve a
Easy
couple of
going and
Niskins to
drop me
sample
off
these
up
later.
Bob's like, you're driving your own dang ROV.
Yeah, so you see it. This is the same, but narrow. So it's like 560 meters. We'll see. So we were looking at this data while the ROV was on deck, so I put it back on so we get more data.
Yeah, okay. So we got another cast through it.
I'm sorry?
Was that down or up?
Both.
Oh, I see. Wait, you didn't see it on the down, but you saw it on the up?
That's right.
What the heck is that?
But this CO2 anomaly, she saw it both in the up and down in both dives.
Oh, interesting.
So,
Bridge nav.
yeah, we want to fire a couple of Niskins at this depth on the way up.
I wonder if we could use the CO2 to determine whether we're in that.
Can we move
layer.
another 30
Yeah.
meters due north, please? Thank you.
So, cool stuff. Unexpected.
Yeah. That's the thing about exploration.
That's right.
Beeping? Oh, interesting. Oh.
I mean, yeah, the closest I was seeing was the bury a day for that sponge. So, yeah, but nothing quite matches up to it in the guides. That's just really an amazing sponge.
Oh yeah, I actually saw that. Mm. Yeah, they've... Right, okay. Oh.
Are those little tiny starfish, the little white things?
Oh. Yeah, I saw what you're talking about off to the right.
They're itty bitty.
Can you zoom in, Dave?
That's a technical term there, by the way.
Yeah, that's a little sea star.
Oh, that's adorable. Okay, thanks. It amazes me how much we can zoom in. It's like better than being there in person.
Now,
Love it.
you're a little quiet on my sound.
I haven't had a chance to fix up the intercom yet.
Oh,
With
okay.
other stuff. I'll get to it soon.
So those little sea stars, they are cute, but they kind of like just munch their way across the seafloor, eating whatever they come in contact with.
This is true. Sea stars are sometimes ravenous beasts. We definitely see that up in the shallows, and I've noted that on a number of the videos I've watched. Just you'll see sea stars up in the corals and just completely bare branches.
30 meters to the north,
It's like a bamboo whip there.
Yeah, and we saw that one that was, looked like it was hugging the sponge.
Oh, yeah.
But it wasn't.
It was eating it.
Oh, that's another predation event for this watch, seeing that.
I should put together a compilation.
Yeah.
All the things we saw.
For Halloween. Gosh, those yellow stalks are pretty amazing.
Do we know what that coral is in the foreground? Is it a bamboo?
Might be Ramilla gorgia. That white kind of branching.
Yeah. Be interested in seeing that sponge a little closer too afterwards.
Oh, and it's got a little feather star crinoid sitting on top of it.
Yeah, a little itty bitty.
I don't see any bamboo sections. But maybe that's just my eye.
And there's a dead sponge right next to it in the front.
Yep.
So it seems like they were like fighting over that territory.
They're definitely in a standoff now. They're like, this is my spot. You move. No, I can't. You move.
Oh, that's pretty. It always amazes me, these bright colors, when there's no light down here.
Hey, bring it on.
Those Bolsonaros, what is it?
Bolosoma.
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 I'm not 100% sure. Still working on my sponge IDs,
But there's a lot of them.
there's a lot of these stock sponges down here. At least 2 different types. There's maybe 3, because there's some smaller, more delicate ones that are all white. There's these ones with the yellow stalks, and there's some with like a very compact, rounded sponge head. Ooh, a sea cucumber. And then there's some with these really big balloony heads. So
that one literally looks like someone dropped like a Japanese eggplant right on the seafloor.
it does kind of. I like this purple color though. It's one of my favorite colors.
Oh, less like it when you get up close. Oh yeah, that's a frilled beauty. That's not the scientific name. That's the common name.
It looks like it's actively feeding. Looks like it has its mouth down.
Okay, so which end is the mouth?
The one on the right.
Okay.
And
Looks a little stopped up there.
It does. So it's probably getting ready to push out some fecal pellets.
Oh, it's kind of cool if you if they get frightened and jump off the bottom, they often
The sea pigs always
drop the ballast weight.
They do that. All right.
It's pretty amazing. Some of these sea cucumbers can actually swim. They're quite lovely when they do.
Swim kind of sounds directional. They can locomote. Oh.
Very cool.
Bridge nav. Hi, can we add another 30 meters due north, please? Thank you.
Oh, I see the smaller, more delicate sponge there
Mm-hmm.
that you're referring to.
That camera is driving me nuts, sir. I think I don't want any up there
So
anymore.
I tried everything I could do and sent Matt a message.
It's very distracting.
Okay, well, I'll put something else in.
Right. If you got a golf tournament or something, you can put up.
Like what? A golf tournament?
Yeah, the cinema camera, that herky-jerky thing is like really driving me crazy.
But why does that remind you of a golf tournament?
No, I'm just saying, well, he does sports stuff, so.
Oh.
He put something else up there besides the herky-jerky cinema.
Oh, yeah, put a... Wow, golf? Oh, boy. That's...
Well, then I wouldn't be watching it.
Yeah, no kidding. But I might fall asleep.
You zoom in, Dave?
Not and fix your problem at the same time.
Well, it's fine if you stop moving.
Yeah.
Oh, interesting. That's, not one that I've seen before. Cool.
It's not a juvenile version of the big dude?
I don't know. I don't think so,
I think
but
it's different. It's
yeah.
got a different, slightly different structure to it.
Yeah, the opening on the top there is...
Sort of looks like a miniature version of the other one, me.
Oh,
Photo bomb. Even down here.
All right, cool. More of that same potentially Ramilogorgia coral.
No, that's not going to work either.
Sorry.
Yeah,
How about a big version of that?
that's great there.
Sorry, Bob, my finger slipped.
No, I like
don't know how
that view.
The first one, though, I don't know how that got there.
Uh-huh.
What's that over there?
I really like how that view makes you look like you're about 400 pounds.
Are you looking right there in the center, Adam?
No, I was looking down on the left, but that's fine. It's like a little cucumber or maybe an anemone.
Oh, I missed it. Oh, well.
We got a long way to go.
We do have a
so
long
it's
way to
not-
go. There will be more to see.
It's interesting. This area doesn't have a lot of diversity. It has a lot of the same organisms. I think as we get shallower, we'll probably see more diversity.
Change nav. Can we move another 30 meters due north, please? Thank you.
Oh, there's a coral whip coming up for us.
Ooh. It's a pretty long one. It's fun with this fisheye lens, be able to see a little bit ahead of where we normally can see. How far up does it go? Oh, gorgeous.
How high are you, Bob?
6 meters?
Wow.
That's a long one.
That's the back, though, so it's probably 5 meter-ish.
Yeah, still.
Very cool. That little colony of corals has been working hard, growing long, that long. I wonder how old it is.
Yeah, who knows?
And how many polyps are on there? Don't worry, I'm not going to ask to count them today.
We could figure it out, right? We just need to count how many are in 10 centimeters.
And then multiply, yeah.
I was told there'd be no math.
It's not math, it's just counting.
All right, let me get my abacus. We can figure this out.
Take off our shoes. Get our toes going. All right, so we do have some smaller whips down there below too, those delicate sponges, the yellow-stalked smaller sponges.
Does anyone even know anyone who knows how to use an abacus? That's like, that'd be a cool party trick.
I know a little bit about slide rules.
Yeah.
I had a really fancy stainless steel slide rule my dad had given me.
Oh, that's cool.
I have 2 of them. I have a stainless steel one, and I have a rosewood inlaid ivory scales and all of that kind of stuff.
Wow.
It was my grandfather.
So how's a slide rule work?
Yeah, I used to know that. Well, back before scientific calculators, when you were in school and you had to figure out equations, that's what it took.
Yeah.
Dang, look at these big.
Oh, those are
pretty. Wow.
Got a little one and a bigger one. Little succession there. I wonder how big they can get before they fall over and break these stalks.
So, I guess they're, oh, there's a
Bracengid up there on the rock. So that's a type of sea star.
We have nice clear water today, Esteban. So.
That's lovely.
Vano, would you make a highlight on the Atalanta camera?
Copy.
Thank you.
There's a coral slightly to our left off screen. It looks a little different than what we've seen. I don't know if we're too far to see it.
Is it up ahead or behind? Where is it?
I can see it in the fisheye to the left. There it is.
Ah, bottle brush Chrysogorgia.
I love Chrysogorgias. They're so delicate and pretty, and sometimes they have little squat lobsters living inside of them. I think this one does.
Can you zoom in, Dave?
I mean, don't we all have a little squat lobster living inside of us?
I hope not.
That's our tells
We have
tells
that
us to
song.
be tells us to be good.
We have the song, right? Every time you hear that word, yeah. Don't you get that song in your head?
What song?
Are you thinking of rock
B-52s,
lobster?
Yeah.
Oh. yeah. Whoop.
Now it's stuck in my head.
Yeah, there's a...
There we go, a little squat lobster blending in so nicely in the colors. There are two of them, aren't there? Three, maybe? No? What is
I
that?
see
Up on the end?
two.
Two. There's something small on the right side. I can't tell.
Yeah, I think it's just a inflamed polyp. But they don't have inflammation problems down here.
It might be
Pretty
splitting though. I can't tell if it's like a polyp splitting or if it's something else stuck onto the branch. But I want to know what those squat lobsters are doing.
They're just chatting.
It was this big, I'm telling you.
Morning convo.
No, it was this big.
Yeah, and so I was following. So I put my hands like this. Woo!
And there was a sea cucumber behind them on the sea floor up to the top left. Another purple sea cucumber.
All right, I'm moving on, or I'm happy to move on. I can't move on myself. That's why I rely on others to help me move on.
It takes a team. You're okay. We're here for you.
I made a breakthrough.
Well, admitting you have a problem is the first step to recovery. Now let's talk about the rocks.
Yes. Thick coating of iron manganese oxyhydroxide. But there's rock hidden underneath.
Bridge nav. Hi Flavio, can we add another 30 meters due north, please?
There's a pink
Thank you.
cucumber.
Pretty good easterly current, or
Definitely.
That would kind of align with the signature they saw from the sage and the mapper, right? Wait, is easterly headed from the east or to the east?
to the east.
Yeah, so that would explain that kind of plume that they think they might have seen with some carbon dioxide higher up in the water column. Or maybe different up there, but...
Well, the confusing thing is that we didn't see anything that high over the volcano itself.
Oh.
So it have to move upwards, perhaps?
So you can see how far Atalanta is in front of the ship, you know?
Oh, yeah.
Huh.
And I'm having to give it full lateral to just stay along the line.
Huh.
Except right here, it just died. I think we're just hiding behind a rock, you know?
Yeah.
It's like when you're scuba diving, you can kind of dodge the currents by kind of getting behind the rocks.
So I haven't seen, I think in the beginning they saw a live version of this Faraday sponge, but so far we haven't seen any alive.
This big one?
Yeah.
Oh.
They saw a big tree?
No, they saw a little version of it. I'd love to see one of those.
Yeah.
Johan, can you direct the ship to find one of those, to the next one of those?
Yeah. We just used the giant sponge odometer.
You turn on the sponge dar.
I'll call the bridge. They know what to do.
Is that a polyopagon there on the left?
Yeah, looks like it. Not super healthy, but...
Yeah.
Yeah, the current's back.
Oh.
So we were just hiding behind that rock. Full beans. You zoom in, Dave?
Yep. Polyopagon. All right, I'm good.
Bridge now.
I just realized I don't think we did introductions.
Oh.
We just jumped straight into sponges.
Can we move another 30 meters to the north, please? Thank you.
You want to fire us up, Fano, so we can introduce ourselves?
All right. Yep. Talof and welcome, or good morning from our end. It's Samoan Standard Time, 8:40 in the morning, and this is the 8 to 12 shift on our Nautilus expedition called E Mamano Ngatai Whalelua. And it's great to have all of you guys here as we continue this expedition, or our dive H2062. But before we get into our details for our dive, we're just going to go round the robin to introduce yourself, your role, and Adam. Do you want to?
No, no one liked my idea.
No.
It's the first thing they've all agreed on is not to do my plan.
Okay, let's see. So our question for today, if we could have any animal as a team mascot on our ship, or, what would it be?
Oh, that's a good question.
A team mascot to represent our team. What would it be and why? Anything. Yeah, so, anyone wants to go first? Any volunteer wants to go first? Or
I'll go first.
Okay.
So I'm Adam Sewell. I'm a proud graduate of Washington Middle School. Go Bulldogs. I'm sitting in the watch lead for the 8 to 12, and I'm from University of Rhode Island. I study submarine volcanism. I serve as the director of the NOAA Ocean Exploration Cooperative Institute. And if we had a mascot, I think 100% it would be a coral, because we are all a bunch of polyps working together to create an organism that functions as one to explore the seafloor.
Oh, nice.
That's a good one.
So Adam, you can choose who
goes next?
Yeah, who goes next? Yeah.
All right, Dave.
Morning, everybody. Dave Robertson, lead video engineer on the 8 to 12 watch, zooming in on things. Let's see, a collective group animal. Well, I always like looking for octopus. And I still hold a hope that we'll see an octopus. On every cruise I've been on, I've seen an octopus on my watch at one time or another. So, hoping for octopus.
Yeah.
Dave, you get to pass it to whoever you want to choose
Is
next.
there 8 of us in here?
Oh, 8 arms on a 8 tentacles
Oh,
on
there
an
you go.
octopus.
Good one, Bob. Yeah, okay, Bob, you get to go next then.
I'm Robert Waters. I'm the herd pilot on the 8 to 12. Proud graduate of Pioneer Middle School.
Oh, yeah. Remember the mascot of that?
They're gold miners, yeah. Like miner 49er. Well, I like the octopus thing, but I can't use that, right?
That's right. No repeats.
Yeah, I guess I'm at a loss then.
Bridge nav.
Okay, eDNA as the mascot.
The Edna crew.
Wow,
look at all these
I'm really
sponges.
struggling to hang in here with the current. It's really blowing. You have to face into it and go sideways.
Yeah, another option is to, like, we're on this ridge. We could get over the edge of it and hide out a little bit.
But then I don't think we'll see all the cool
Thank you.
stuff.
I could probably, we could do it this way, I think.
Okay. All right, Bob, who's next?
Did Johan go yet?
Nope.
I have not. Hey everyone, my name's Johan. I'm the navigator on this 8 to 12 shift and on the boat. Off the ship, I'm a PhD student at the University of Rhode Island. Go Rams.
Hey.
And I'm going to go with the obvious mascot choice. I think we should have a Nautilus.
Oh, interesting.
Always wanted to see one.
Yeah, no kidding. Apparently they're in this area. Is that right, Val?
They are, but they are depth limited, so they can only go down to about 800 meters. Below that, they'd crush their shells. So they are a little more shallow, but maybe when we go to some of the shallower ones close to the end of this expedition.
What about if they died, would their shell survive falling to the seafloor, or would it still crush? Is it like
I think
it's got implodable volumes in it?
You know, I think if they died, it might be okay because they're controlling the pressure inside the shell for their buoyancy and stuff.
Oh,
so
really?
I
At
think if it
800
fills,
meters? That's
yeah,
amazing.
if it fills in with water, then it wouldn't crush. So we have found shells, like Pisces found shells, found a shell in Whangatele Bay actually during their 2005 dive there. So they are around. I think mostly you can see them with baited cameras. So that's been the method that they've used here, is to send down a baited camera to attract the Nautilus in. And there's some really funny videos online of the Nautilus here. They're just bumping into each other. Maybe we could drop some bait at one of our waypoints.
I don't understand how they can maintain buoyancy control at 800 meters. That sounds...
Right?
That's some pretty high pressures. Like, how does it do that?
Magic.
Yeah, I don't know.
And that's like titanium kind of pressure vessel kind of.
Yeah.
All right, so
Reset.
Johan, pick who's next.
I'm going to popcorn it over to Erica.
Hi, my name is Erica Leon Guerrero, and I'm an ocean science intern. On this shift, I am a data logger. If I could choose any mascot, it has to be something we've only seen once this dive, and I think it's gonna be that translucent spotted red squid.
Oh, that was so cool.
It wasn't a squid, though.
It wasn't?
No, Megan said it was octopus.
I thought that might be
okay.
the case. Awesome.
Okay, then forget it. It's not our mascot.
Well, it's already been said.
Yeah, that's a
You
really
can't use octopus.
Okay.
We
I'm
have
stumped.
seen other squid.
No, I really like that Swifty squid.
The Swifty
Yeah.
squid.
I will give it to Funnel.
All right. Yep. My name is Fano Manga. I'm from American Samoa, teacher in Untitled Elementary School, and currently I'm SCF on this 8 to 12 shift. And if I want to have a mascot on our vessel, I would say I'm just gonna go with something different, like an ROV.
Oh.
Yeah, with the two manipulators can be the hands,
Yeah,
and then that big
fight
camera.
ROVs.
Yeah, on the top can be the face, the head.
bridge
Yeah,
now.
yeah.
All right, who you sending it to?
Yep. Aspen. Yeah.
Well,
Can we move
you
another
guys
30
left it
meters to the north,
a little
please?
difficult, but I guess I'd follow up with Fano's suggestion, and I'd add like a sword, and I'd do like a whole Roman theme with Herc, Argus, and Little Herc. Do the whole team up. Yeah, those would probably be the mascots I choose as well.
Nice. And who the heck are you?
Yeah, and my name is Esteban Juan. I am an ROV engineering intern from the University of Puerto Rico at Mayagüez. I'm a graduate student in the oceanography. And yeah, I'm sitting in the Atalanta pilot seat for this watch. So, yeah.
All right, Val, you got to close us out.
Oh, it's me. I get to go last. Okay. Talofa, everyone. My name is Val Brown. I am the research coordinator at the National Marine Sanctuary of American Samoa, which is where we are at right now. So very exciting. If I was going to pick a mascot, I think I would go with our humpback whale friends, because a lot of people here have traveled very far, but we all know how to have a little fun.
There you go. That's a good one.
All right, there you go, folks. Again, the chat on nautiluslive.org website is open. If you have any questions or comments, just put it in the chat box, and then we'll be able to help you out with your questions. All right.